Hey, it is Mr. Wokenfeld. Welcome to another AP Bio review session. We're going to do some biomania. We're going to do some music. But first of all, I got to know who's here. And so what I'd like for some people to do is to just sort of say hello so I can see some people in the chat. Want to see who's here. I've got some people who have become regulars for these events. And I got Matthew Diggins here. I've got, Sumak Sundaram. That's fantastic. How about a couple of other people? It is bio time. As Matthew was saying, that is making me super, super happy. I'm wondering if we're going to have anybody else come here because, the more students we have here, the best. Matthew, you come from a small town in New York. All right, that's fantastic. Biggie Cheese is here. Way to go, Biggie. Marion, Sumik. All right, this is fantastic. I'm seeing a lot of people come on here. This is great. Hey, why doesn't everybody do this though? Why doesn't everybody do the kind of thing that your teacher would hate in the classroom, but which I'm going to love? And that is, I want you to take your phone and I want you to text 5 people and tell them that Mr. W is doing a live AP Bio review session. A live AP Bio review session, and he is ready for hundreds of people because he wants to do it. All right, I am so glad to have people here from all around the United States. This is making me really, really excited. I'm doing a little bit of adjustment of some stuff here just so I can see all the things that I want to see on the screen at the same time. Pretty good. I got this pretty good. I got Sophia. That's fantastic. Hello, Sophia. How are you? It is great. Yeah, people are telling me where they're from, and the idea that I've got people all over the country who are listening to us in this difficult time, right? This is a difficult time to learn AP Biology to get ready for the AP Bio exam, but we're going to do it. We got a lot of time. There's just so much to learn. We can take it nice and slow. And the thing that's a little tough for me is that we've got this little chat window. I know that it's a little asynchronous. I know that you're out there trying to communicate with me and it comes in a little bit late. And again, I'm wondering if we can get our numbers up a little bit. I'm going to check over here. We've got 12 concurrent viewers. Oh, now we're up to 13. That is very, very exciting. I'd love to get this number really high. Omar, how you doing? Great to have you here. Has everybody done what I've asked? The forbidden thing in school? Oh my goodness. I've got someone from India who's here at 2:33 a.m. That is incredible. Incredible. Matthew, thank you so much for that comment about my flashcards. That is really great. I worked on them for a really, really long time. Okay. So Michelle, that is very useful feedback. Let's see what I can do about that. Okay, I'm going to go to my settings. Michelle, that is fantastic. All right. Let's see, what can I do? Hey, Emily, fantastic. Emily, how's my volume there? How am I sounding? Am I too low? Can you hear me okay? Give me some feedback. I've heard a little feedback on the sound. I think that was a little bit of my problem last time. Still figuring out some of the settings on this streaming thing. It's a lot easier just to be in the classroom and to talk loudly. So let's see. I can bring things a little bit closer to myself. There's a little danger in doing that. And of course, you know what the difficulty is? It's very hard to buy any equipment for streaming these days. To buy a webcam, to buy a microphone, very, very difficult things to do. So anyhow, Michelle, you got me turned up all the way. Sound is fine. Emily is good. Sophia is good. All right. Okay, good to know. All right, fantastic. So guys, I don't know if we really have any. Okay, good. San Giovanni, I'm not sure what I can do because the sound thing's a little bit complicated, but turn up your volume and we'll do as best as we can. I'm going to do a check over here. I've got 15 people viewing now, and that is fantastic. And you know what I thought we would do? I thought that we would start with a song, all right? So here's what I want you to do. You guys should be looking at my website. I mean, you know, I'm showing you my website. I don't know what you're exactly looking at, but here's the idea. So if you're in my website, if you go to contact, etc., all right, go down here to music videos, all right? And then what I'd like you to do is I'd like you to bring up Osmosis. But here's the deal. Don't play Osmosis, because I'm going to play it out of here. And we're going to do a karaoke version of it. So we're all going to try to sing it together. And this is challenging. The lyrics come really fast, but this is a great way to review because who knows? Who knows what's going to be on this AP Bio test that you're going to be taking on May 18th. But is it possible that there might be something about osmosis, you know, like in the 2018 question number 2, there's one about these pores that open up in the cell. This is sort of a mechanism for cells to actually fight off bacterial infection. How do they do it? They do it by osmosis. So knowing about osmosis, that's a good thing to know about. So let's go. You guys ready to do some singing? All right, let's go. I'm gonna hit it and let's hope that you can hear something. Okay, first of all, I had some sound problems last time. Just in the chat, tell me right now, can you hear the music? Could you hear the music in the chat? Just give me a yes if you could hear some of that music. I'm waiting for somebody. I'm going to take a little sip of tea. Anybody? Could you hear that music? Could you hear the osmosis music? I started to play osmosis. Huh? Yes! Fantastic. Let's hear some music. Let's do some singing. All right, I'm going to go like this. I'm going to go like this. And let's sing Osmosis. You guys ready? Let's do this. I took them to a science experiment. Cell in an environment that's more watery than the cell is inside. What I saw came as no surprise. My gummy expanded, quintupled in weight. My gummy expanded and increased so great. So listen up as Mr. W explains this. It's all about osmosis. Osmosis is the diffusion of water across a membrane or a water permeable border, diffusing like our molecules in fluid situations from higher to lower concentration. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. In osmotic terms, hypotonic, hypertonic are the ones that are going to work. Just water's watery diffusion. Learn them well to avoid confusion. Hypotonic means higher water concentration and relatively lower solute concentration. Hypertonic is the opposite, percent of water is less, with more solute dissolved inside, as you might guess. So let's take these terms and apply them to our gummy bear. I'm looking at the package and seeing what it says here. Bear is mostly sugar with some other stuff mixed in, and holding it together is the protein gelatin. So when you put a cup of H2O, it's apparent that this gummy is so hypertonic to the water that it's in, and the water is hypotonic to our little gummy friend. And so the water will diffuse into the gummy, which soon will look so huge. The water which will infiltrate the gummy, which later is looking pretty funny. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. This plant name is Elodia. I never in the salty sea. For the Elodia cells are full and firm. It's all about osmosis, as we're going to confirm. See, the cells are hypertonic to the watery exterior, so nothing could diffuse to the cell's interior. Membrane, pushing it against the wall, so you can't see the membrane at all. But add some salty water so the outside's hypertonic. Hypertonic cells are so osmotic. Water leaves wall, the cells inside are scrunched, so the cells are... Hypertonic brew. Water in the veggies look brand new. Preservation is motto. Kind of makes it right for hypertonic. All right, guys. I think I gotta stop this. I'm screwing this up. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic. Oh my goodness. Osmosis. Guys, that was a disaster. You know what I learned from that? I learned from that I need to practice like crazy to be able to pull off a live version of these. And here is my promise to you. I am going to practice. I am going to practice really hard. And so the next time I do a live performance, in fact, it's going to be Osmosis. Next time we're going to be Osmosis, and you're going to see me nail this entire thing. Because what's more fantastic than doing karaoke biology? And I actually want to let you know, on my website and on my YouTube channel, you can do your own karaoke biology, and there is no better way to master the details. Now, no promise is being made that if you can do karaoke biology, you will do well on the AP exam. Those are pretty different skill sets, but at the same time, for you to master biology content, karaoke biology, these songs in general, are a really good way. So I totally want to encourage you to do that. And in fact, you can have like virtual karaoke parties. People can be doing stuff on TikTok, or I don't know, whatever you guys do. I tried to do TikTok. I didn't really get it. I don't know why. But anyhow, I'm doing this, and this is what I can do. So anyhow, Osmosis, sorry, that was kind of a disaster. But you know what I think I'm going to do? The song is pretty great. So let me actually pull up the song. There we go. And let me go to where we left off. Here was this. Okay, here we are.
Water in the veggies look brand new. Dried fruit and beef jerky preservation is osmotic. Their low water content makes the dry food hypertonic. So any germs that mold fall upon them lose their water and die and don't contaminate the dry food in the larder.
Osmosis.
H2O diffusing.
Osmosis.
See the water oozing.
Hypotonic to hypertonic flow. Osmosis.
Let's go. Animals, cells and situations hypertonic, lose their water, shrink and shrivel. Looks so sick, but an animal cell in hypotonic abode gains water and expands, eventually
Okay, I wanted to show you that because that actually shows up on one of the AP essays. So I want to just point out, there's a lot of content, a lot of really important content. And you know what? Matthew just asked me a great question. And what I'm going to do is this. I'm going to go back to this PowerPoint, and I want to show you something. Just want to remind you guys, the best way to review is to be using my stuff, the stuff on my website. I've got FRQs, I've got flashcards, I've got multiple choice questions, which are also great for content. I also want to emphasize that what you want to do is just make sure you're only studying units 1 through 6 and study the stuff on the College Board website. The main thing is that, I know that this is a little paradoxical, I'm doing a video, but like doing videos, reading your textbook again, that is not the way to study. The way to study is to be actively studying, to be doing FRQ after FRQ after FRQ. So now, what I'm going to do is I'm going to pause a little bit and what questions do you have? So Matthew, this is for you. Matthew, can you go ahead and anybody can actually ask me some questions, because I want your questions to actually drive a lot of what I'm doing. I mean, I've got an FRQ that we're going to do together, but if you have questions, I want to hear what they are. So Matthew, type away. What's your question? Anybody? Anybody got some questions? Okay, somebody? Here, you know what, I'm gonna maybe stimulate you. Okay, everybody see that in the chat? I am now open for student questions. I know that there's a little bit of asynchronicity, but let's hear what some questions are. And Matthew, I just want to let you know, I certainly do have a plan for what we're going to move forward with today. I've got an FRQ that's waiting to happen, but let's see some questions coming on in the chat. Anybody? Okay. Okay. Faith, what's the difference between explain and describe? All right. So, first of all, one thing that I want you to know is that there's a document that you totally have access to. It's the AP Bio exam course and exam description. And in fact, let me just show you guys this over here. Pop over here. So everybody should be looking here. And I'm going to go not here, and not here, and not here, and not here. Well, let's go here. AP Bio course and exam description. Okay, so here it is, first thing that comes up. And here's the thing that I want everybody to know. This is not like a secret document. This is available to everybody. And in this document, you can get an explanation of what all the task verbs are. I think like they're on page 197 in this document. So in terms of Faith's question, Faith, go ahead, look through this document, find the task verbs, and you'll see a general explanation of like what's the difference between explain and describe, predict, justify. These are the kind of things that they're going to ask you to do. I think they're, for the most part, pretty straightforward. But Faith, that's what I want you to do. I want you to go look at this document. Okay, Matthew, you're more than willing to do countless FRQs, but I really don't know how to start. This is complicated, Matthew. On my system, so if we go to Science Music Videos, which I have right here over here, I don't need to type it in. I want to really encourage you as much as possible, Matthew, to really work through the FRQs and the flashcards, maybe mix it up a little bit. You've got to know the content, but you've also got to set yourself up to be dealing with specific kinds of challenges. I try and do that as much as possible on my flashcards, but I also want to be really clear. The best stuff to study is this stuff right here. So for example, if you go to past AP Bio questions, okay, you see right here? This is it. This is the best. So, you know, here you've got all the free response questions. And I actually have a separate document where I tell you what to do. Like, for example, this question, this is right in line with what you're going to have to know from the AP Bio, you know, for this coming AP Bio exam. You could get a question just like this. There's no reason why this isn't fair game. So that's it. So Matthew, you got to work it all. Work the flashcards, work the FRQs, work the multiple choice quizzes, but the thing that I mainly want to tell you to do is make your learning active. Active learning. That's what it's all about. And so that's why I want you to quiz yourself. And in fact, if you are really behind, then just do this. So go to my website. Let's say that your teacher didn't get you through all the way through number 6. Well, I've got tutorials pretty much on the entire curriculum. And so you just go through and, you know, you can skim the reading and then just go to the quizzes. And the quizzes are going to give you tons of feedback. So we're just going to close that. And You know, you'll be told if you get it wrong. And, you know, it's a sort of a more painful way to learn, but this is the way to do it. Don't be passive now. Make it hard. It's difficult learning that's going to lead to big results. And so I totally want to encourage you to do this. Okay, so so many questions. And I'm going to take one from Emily. Emily, who is my student. And so Emily, what does the N-terminal mean in terms of amino acids? So amino acids, remember, like there's a central carbon, and then there's a carboxyl group on one side, and there's the amino end on one side. So one side of a polypeptide chain has an N-terminal amino acid, and the other side has a carboxyl. And they're just telling you that it's at the end. That's really all that it means. And it's not different from a normal amino acid. It's just indicating that first or last. And, you know, directionality is important. I don't really think you need to trouble yourself about that too much. Cherry is asking me to do a video about embryology. Cherry, I'm not going to do a video about embryology because embryology is not going to be on the AP exam, and I'm keeping things extremely narrow. So no embryology. But there are principles of development that are important. So like, for example, if you go here, sorry, just lost my screen for a minute. So you know, we're having some big trouble here. I can handle this. Okay, so if we go here, and if you scroll up, AP College Biology. So if you go into Unit 6, you see this stuff about animal development and cell specialization. That's important stuff. It's very detailed. You know, this is a very constrained AP exam. Nobody knows what's going to be on this AP exam. But Framboise, don't worry about embryology, but you might want to do this tutorial about the principles of development. You know, all of these might take you a couple of hours. You know, biology's hard. You might have to dig in and do this, but like, do it, okay? I want to take a look at something, guys, for just a second. All right, fantastic. All right. Okay. Sumuk, I'm probably not saying your name right, but listen, biology's hard. It takes a lot of work. I make my students work so hard, but there's a huge payoff from it, which is that they gain incredible levels of mastery, and my kids generally do great on the AP exam. So Sumuk, just take it slow, go through the material, go through my tutorials. Just focus on units 1 through 6. You actually have a lot of time. And I know that this sounds crazy, but like if you spend, this is going to sound crazy, but like if you spent 2 hours every day between now and May 18th, you would learn so much. And like, make it your hobby. I'm not saying that like things aren't incredibly stressful now. I'm not saying that the pandemic isn't terrible. It's terrible. But a lot of us have time. A lot of us don't. A lot of us have family responsibilities, and my heart goes out to you if that's your situation. But you can probably squirrel away. I don't know why they say that, squirrel away, but you can probably steal away with 1 hour on the computer to do the tutorials on science music videos and do the reviews. Yeah, and Sumuk, I'm just going to tell you, you just got to take it slow. Flashcard by flashcard, FRQ by FRQ, you're going to get it eventually. So Omar, you're asking me about past videos where I worked through past FRQs. Not really. And the reason why I don't is because all of that is College Board stuff, and I don't think I have really like the copyright to make a video about that stuff, though I have asked. And I'm going to take this question from Grace, which was, how would a cell having pores affect its osmolarity? You know, it's not so much that a cell having pores, I guess I can actually show this. I'll bet you this is fine for me to do this. So here we are. I was showing this over here. All right, so here's a pore. in a cell. How would it affect its osmolarity? It doesn't affect its osmolarity. But usually the extracellular environment is different from the intracellular environment, and usually the extracellular environment is more watery. So in this situation, this is actually part of an immune response where this activation of this enzyme called caspase causes this protein to become active, and that opens up pores in the membrane, and that allows water to diffuse in. That winds up killing the cell. And that might sound like it's a bad thing, but it's actually a good thing because this cell is trying to protect itself from infection. Anyhow, just doing that made me a little bit nervous, so I'm going to stay away from that. Flora, can you explain redox reactions really quick? The reducing agent in a certain... So, Flora, that's really deep chemistry, but here's the basic thing. In cellular respiration, what happens is that glucose gets oxidized and oxygen gets reduced. When something picks up hydrogen, all right, it winds up picking up, it winds up getting reduced. So water gets reduced during cellular respiration and glucose gets oxidized. Think about what happens to the carbon in glucose. It starts out as glucose, C6H12O6, and then it goes down to CO2, oxidation, all right? So that's basically what you have to remember. And then on the other side of that, you have to remember that in photosynthesis, carbon dioxide gets reduced to glucose or some other, 3-carbon intermediate, and water gets oxidized. Now, there's a lot of things that happen in between. So like, for example, NADH, you see that H at the end of the NADH? That's the reduced form. It becomes oxidized to NAD+. NAD+ gets reduced to NADH. So what you need to do, Flora, is I'm going to show you this. I'm going to jump over here. Let's go to Science Music Videos and see where it says AP College Bio. If you go here to Cellular Respiration, in one of the tutorials here, I give a nice overview of like oxidation and reduction and what happens. So that's what's going on. So let's see, how else are we doing over here on questions? All right, so Flora, Grace, all of you guys are going to do the cellular respiration tutorials on my website. And again, here's the thing, if you're part of a class, the website access is free. And if it's not, I sell it for practically nothing. It's $19 for a subscription to Science Music Videos. And if you really need the tutorials, you gotta get the website. If you just want to review for the AP exam, you can get the app on Apple or Android. It's $9.99, I think. So go ahead and do that. All right. So I think what I want to do is this. I think we're going to move on to some review. All right. So here we are. Let's go forward. And I can't resist doing this because I love it. Max Cowan is an incredible musician, and I'm singing on this. He took my voice and he did this. Listen to this. Oh no. Let's try that again. I love that. I can do it all day long. I can't believe he took me, he auto-tuned me. Is that fantastic or what? So guys, let's do some FRQ review. So guys, I want to emphasize, just want to show you one more thing. I don't have infinite capacity to do this, but I want to point out there's a contact button over here on my website. And you can send me, you can send me an email. All right, so there it is. Brings up, but I'm not going to be able to respond to thousands of emails, but go ahead. But the main thing is that, like, if you're stuck on content, Science Music Videos has a tutorial that's going to get you through it. And don't read the textbook. What I've got is orders of magnitude more effective than the textbook. This is the quickest way to learn. You can sort of beef up where you are on content, and then you can go ahead and do that. And all you guys, that is so great. I'm hearing all these great things. So let's do this. Here's my PowerPoint. We're not going to do this again as much as I'd like to. Okay, so here we are. All right, so guys, here's what I want you to do. You can read this on the screen right now. Of course, you can't read it on the screen right now because I'm in the way, so I need to go to my, there we are. There, that's perfect. Okay, so here's what I'd like you to do. I would like you to take a minute. I want you to read over this FRQ. This is an original FRQ. This is from my BIOMANIA system. But I want to point out that if you need to, you can go over here. Science Music Videos, go to Mr. W Live, and I put the same text if you're having difficulty reading this on screen. So everybody, take a minute, read this on screen. It's actually bad for someone to read and for you to listen. Think about the answer. I'll read it silently while you do the same thing. Guys, I'm so sorry. I made it go away. Now it's back. Okay. So guys, everybody get to a good stopping point in the reading. Kind of weird. All the things that I do as a teacher, I can't really do right now because there's such limited feedback that's coming back from you. But basically, here's what I want you to note. Over here, we've got a receptor. Now, this is not like half a receptor. It's the whole thing. It's a real functional receptor. It can bind with a ligand. A ligand is anything that binds with a receptor. So this is capable of binding. But this is the monomer form, and this is the dimer form. So you got a couple of questions that I want to walk you through to get to some good understanding of this. And one thing that I want to emphasize is that on the AP test, both questions are going to involve predictions and justifications. And I'm going to try and model that in the work that I'm doing here. So, did that do anything? No, but now it did. Okay, so here's what I want you to do. I know that there's a big delay and I know that everything's asynchronous, but can you go ahead and can you please do your best to answer these two questions? And you know, when I get 10 of you to type something in there, I'll know that it's time to go on. So, best as you can, what's the difference between a monomer and a dimer? And then what's a disulfide bridge. Okay, so everyone's typing in the chat. Let's see some stuff happen. Okay, I'm patiently waiting, working with the asynchronous nature of YouTube live teaching. Okay, I got something from Sophia. Good. Macy, fantastic. Grace, keep them coming, guys. I want to see 10. Okay, how about the second part of the question? Oh, I can't really do this. Okay, guys, I'm seeing a lot of great answers. Okay, I'm going to go on, and here's what we got going on here. So, monomer, 1 unit. A single amino acid is 1 unit. When that single amino acid gets hooked up to another amino acid, it's a dimer. All right, 2 units. Same thing happens with monosaccharides and disaccharides. Okay, so monomer, dimer. Now, a disulfide bridge it's one of the bonds involved in tertiary protein structure. Now, again, let's think about the AP exam. You're not going to get any little multiple choice question asking you like, which of these is a disulfide bridge? It's just not going to happen. There's only 2 FRQs. But you will need to know stuff about the general principles of protein structure. Or you might, again, who knows? They're going to have to choose 2 topics from the whole AP curriculum. So let's talk about this a little bit more in detail. All right, now, let's review protein structure just a little bit. So in the diagram on the top, you see this is A1, A2, A3, A4, A5. So that's a chain of amino acids. It's a polypeptide chain linked together. That's primary protein structure. That's genetically determined. DNA is telling RNA to tell a ribosome to hitch together proteins amino acids in a certain order, and that's the order that emerges. Now, those amino acids start to interact with one another, and that creates a secondary structure. And those are interactions that happen between either adjacent amino acids, that structure is called an alpha helix, or between ones that sort of get close to one another in a cross-linking thing. With my students, like, you got a polypeptide chain and they can be parallel or anti-parallel, but the amino acids that get close to one another will form these kinds of bonds over here. And note that these bonds are always hydrogen bonds. Okay? And I'm seeing, Grace, that was a fantastic comment about helical things or pleats. Now, tertiary structure is where the fantastic complexity of proteins comes into play, because that's when you start to have bonds between the side chains of amino acids. And that's where the really interesting chemistry and really interesting shape emerges. And then what you can have is aggregation of entire third level proteins, tertiary proteins, and that leads to what's called a quaternary structure. When you learn about this stuff, quaternary is almost always taught in relationship to molecules like hemoglobin. It's a very commonly taught quaternary structure. Now, let's see how you guys are going to do on this. Guys, very quickly, in the chat, can you please type which number over here represents the disulfide bridge? There's a big hint. See if you can do it. I want to see some numbers. Flora, I'm going to get to your question in a minute. First, respond to this that's on the screen. Okay. Grace, good, good, guys. You got all, I got this. Okay, so this is the disulfide bridge. This is actually the end of one of the side chains of one amino acid. It has to be a cysteine, and here's another one. All right, so here's the amino acid, here's the side chain over here, and that SH, that's the basis of the disulfide bridge, because when two cysteines get close to one another, those sulfurs will bond with one another, and that's a very strong, heat-resistant, covalent bond, and that really stabilizes the structure of the protein, and it's causing the protein to bend and turn in all kinds of ways. Now, here's alanine. And in this question, basically, what you're posed with is there's a substitution that causes alanine to be replacing cysteine. So how, bunch of you, type in, just wondering what happens here. Okay, what would happen if there was a mutation that caused cysteine to be replaced by, one of the cysteines to be replaced by an alanine? And, you know, Deborah, these questions are coming fast, but disulfide bridge, it's one of the things that give a protein its tertiary structure. So like, for example, that's a hydrogen bond. That's also giving the protein its tertiary structure. This is called a hydrophobic cluster. That's also part of it. This is an ionic bond. But this is a particularly powerful kind of bond. But it's only one of 4 possible tertiary interactions. And Flora, you asked a question about where the hydrophobic interactions are in the tertiary structure. This is a hydrophobic cluster in the tertiary structure. Okay? So, guys, Okay, so, guys, I'm going to actually grab some text here. You know, this is probably a waste of time because you can probably see it, but look, okay? So, here's what one of my students just put into the chat, and I'm just copying it. So there's no longer a second sulfur atom on the side chain, so the ligand binding domain can't form a disulfide bridge, and the ligand can't bind. So guys, look over here. If there wasn't a second sulfur, then this structure couldn't form, and that would change the shape of this ligand binding domain. A domain, that's like kind of a fancy word, like the domain of the king or something like that. So basically, it just means the area. And this ligand binding area is now, it's a different shape. And if it has a different shape, it's not going to be able to bind with the, it's not going to be able to bind with whatever ligand it bound with over here. So that's the first part of the question. If you have a mutation that changes the structure so that there's not a second cysteine, but it gets replaced by an alanine, then you won't really have the kind of ligand binding to the receptor that you previously had. Now, I'm going to go to the next slide over here. So here's the deal. The way that this kind of receptor works is that once the ligand binds over here, then this monomer form of the receptor finds another monomer, and they form this dimer form over here. So they're both holding on to the same ligand. The different ones work differently. There are some that I believe hold on to like 2 different ligands, but the point is that they're holding on, and notice this over here. You know, activation and phosphate groups, those things in biology go together like hand and glove. So after the receptor is activated, what do you think is going to happen next? All right, I want you to speculate a little bit. Ah, Grace. You're killing this. It's fantastic. Anybody else? Anybody else? And Sumak, I'm probably mispronouncing your name, and I want to apologize for that, but I want to say that for someone who was just being so unconfident, that was a fantastic answer. So you did a great job. Flora, very nice. Okay. And anybody else besides Grace want to respond to this? Grace is adding even more. Fantastic. Let's hear some more, or let's see some more. Give me some more in the chat so I know when it's time to go on. How about 5 or 6 more answers? All right. So I'm seeing, again, another great answer from Sumuk. There's another signal transduction pathway. So you guys are totally getting this. So basically, what could happen next is this. You've activated this receptor. You can tell by the messengers, by the phosphate groups, and that is going to be activating some kind of cellular response. And in general, there are 2 cellular responses that happen. One is that there are internal enzymes that are in an inactive form that are ready to go. And the other is we're going to turn on genes. So those are the 2 things. And people like Omar and Sophia are just, you're totally nailing this. So you guys are doing a great job. And let's go on now. So I just sort of started from this diagram, and I developed a system where you go from this to this to this to this, and there's a pathway that links the active form of this receptor to the cellular response. I need to move myself to this side of the screen. Here I am. What do you call that pathway? People already said it. I just want one person to put it in the chat. This pathway would be called a okay, waiting for some responses. What you would call this pathway. And you would call this pathway a, you know, people already put it in here. So I'm just going to assume that people have already anticipated the result. And the answer indeed is a, oh, it's all coming in right now. I was just being a little impatient. Yes, it is a signal transduction pathway. How did I get so lucky that even through the internet, I have such fantastic students? Okay, you guys are killing this. Fantastic. So let's pull this all together. Here's what I want you to do. Predict the effect of a mutation in the DNA for the receptor that resulted in changing one of the cysteine amino acid residues into an alanine. Justify your prediction. You see that word residue? Do not worry about it. A residue is just like the amino acid that's in the polypeptide chain. It's sort of a useless chemistry term, but good chance you'll see it because the College Board seems to like using that term. So I need to move myself to over here. No. There we go. Okay, so here's what I want you to do. There are a whole bunch of terms here, and what I want you to do is put your answers in the chat separated by commas, and I just want to see if you get this. If the mutation changed one of the cysteines to an alanine, then a blank blank could no longer form. This would change the blank. I'm not going to read this anymore, but you guys are going to read it and you're going to type it. Go ahead and do it. Let's see some answers come in. Okay, seeing these answers starting to come in, separated by commas, just the way I asked. Good job, Grace. Samuk, nice. Let's just see 2 or 3 more. Okay. All right. Guys, you're totally getting it. I'm going to show you the answers. All right. So, if a mutation changed one of the cysteines to an alanine, then a disulfide bridge could no longer form. Disulfide bond is fine. This would change the shape of the monomeric form of the receptor so that it could no longer form with the ligand. As a result, the receptor couldn't form into its dimer form, which is the active form. And again, you can tell that it's the active form by the presence of these phosphate groups over here. That's the, phosphate is like the activating thing in biology. This would prevent, some of you wrote inhibit, that's a perfectly great answer, whatever cellular response occurred after the binding of the ligand from happening. Okay, so that's it. There it is. You can go back and look at it on YouTube. There it is. So now, predict how this mutation would affect differentiation in cell type R7. Justify your prediction. Now, you could go back all the way, here and look at the original text of the question, right? But all you need to know is that it says over here that this receptor is required for the differentiation of a particular cell called R7. Okay, so I'm going to go forward, 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 forward. We did this. Okay, so what would happen for differentiation of this particular cell? Could you guys please go ahead and type that in the chat? I want you to formulate an answer. How would this mutation affect differentiation? Let's see a couple of more answers come on in. Guys, you got it. Now, let me show you what I put here. And I know that this is kind of a tough context, but what I'm noticing is that most people did the prediction, differentiation of the R7 cell would not occur. And what you didn't do is you didn't do a justification of your answer. So this is just practice. Don't worry about this. But the point to note is that you got to justify your answer. So if you say differentiation is not going to occur, you got to say why. And that why justifies it. And basically, because the receptor is required for differentiation, then differentiation would not occur. That's it. That's all you've got to do. That's what the justification is. So, guys, here's the thing. You should go ahead and, go do something else, clear your mind of biology, but then come back to this question and answer it again, answer the whole thing. And in my BIOMANIA app or on the website, you can compare your answer to a sample answer of mine. And, you got to keep working this until it becomes second nature for you to jam these things out. Biology is a huge topic, but it's finite. So if you keep on working the College Board questions and mine, and the stuff your teacher is giving you, you're really going to get the set of all the possible things that you can get asked. So Sophia's asking, would that sample response be sufficient for an actual exam? Yeah, you know, I've graded these things. I don't know what's going to happen this year, but like I think this would count for prediction, differentiation of the R7 cell would not occur, and a justification, because I put because the receptor is required for differentiation. That's probably sufficient. You might want to elaborate on that more, and I would totally encourage you to go ahead and do that. So elaboration is good, unless you say things that are wrong. It's sort of a double-edged sword here. But go ahead and, you know, make sure that you've justified things. So Sophia, I hope that is satisfying answer. So guys, we did a lot of questions at the beginning, and we're close to the end of our hour together. So you guys want to do a quick quiz? All right. So I'm going to show you some quick questions, and then I want you to put your answers in the chat. Is everybody ready? Everybody ready? Let's go. First question. Mutations in DNA regions that don't code for protein can be silent with no effect on the organisms. Matthew, I think the answer is yes. Okay. Okay, guys, you're mostly right. It's not genotype, it's phenotype, because there's a mutation. A mutation means that we've changed the genotype. We've changed the underlying genes. But what we're not changing is the phenotype. Okay, so that's the first answer. All right, let me go here, here. Okay, next question. Messenger RNA specifies the order of blank blank in a polypeptide. What's the blank blank? Go. All right, very nice. Good job. I'm seeing a lot of amino acids coming in as an answer. Okay, and the answer indeed is amino acids. Let's go to the next one. Okay, gotta say, you're not going to be asked a question like this on the AP exam, but to be a literate person who understands biology, you should know the answer to this. So, X-linked disorder in which blood won't properly form clots so that bleeding stops. You know what I meant there? There's no clotting, so bleeding won't stop is really what I meant there. What's the answer? I'm noticing that it takes about 20 seconds. Nice. It's coming in. Coming in. All right, let's do another one. Here's my cursor. All right, here it is. Answer's hemophilia. Fantastic. The love of blood. But people who are hemophiliacs, they don't love blood. They probably think blood is a drag because they bleed all the time, or they used to. Now it's a very treatable disease. Okay, in this viral life cycle. A virus injects its genes into a cell and then uses the cell's metabolic machinery to turn the cell into a virus factory. What's the name of this viral life cycle? So, the answer is, this is the lytic cycle. All right? SARS-CoV-2 is a lytic virus. It's just injecting its genes, and then it's taking over the cells, and it's using the cells to make more viruses. Lysogenic viruses do something different. Lysogenic viruses insert their genes into the nuclear material of the host cell. So if the virus is just injecting its genes and then immediately uses the cell's metabolic machinery to turn the cell into a virus factory, it's a lytic virus. Okay, guys, we're going to skip these. And here's what I want to say. We're going to close up. I want to congratulate you guys. I want to say, way to go. You guys are awesome. You're here. You're learning biology. And I just want to really encourage everybody that you can get over here to a 4 or a 5. But you have to practice a lot. And you have to have your learning be active. And that's why I'm really promoting the use of my website, which you have to subscribe to, and my app, which you have to buy. And they're both totally reasonable in terms of price. So if you're not in a class that's using these materials, I really want to encourage you to go ahead and buy a subscription to the app, buy a subscription to the website, or just, download the app and do an in-app purchase. Want to emphasize that your practice should involve 3 things. You want to work those College Board questions. They're the best. They're unmatchable in terms of their quality, and that really is going to help you simulate what you're going to experience on May 18th. Then BIOMANIA, FRQs, flashcards, and multiple choice, just to get your content knowledge to a really high level. And then finally, if you want to, and you want to do this work on your phone, you can do this. So I just want to encourage you to do 2 things. I'll be back here next Tuesday. I'm totally enjoying these. It's really fun. We'll do an FRQ. We'll do some music. We'll do some quiz questions. That's going to be our regular thing. And you can help me by getting all of your friends to join us, because it would be really satisfying for me if, like, for example, I was doing one of these and there were and there were like hundreds of people there. So I just want to respond to a couple of questions. Samuk, if we start getting a ton of people, and I think that there's a huge demand, like, there's more than 100 people at each one of these sessions, I'll think about opening up a 3rd one so that people can do it. But, it takes a lot of time to prepare for me. So I just want to make sure that the energy that I'm putting out is going to be matched by people there. So no for now, but get a lot of people to show up on Tuesday, 2 p.m. Pacific time, and, get those people there. I'll be totally happy. You'll be totally happy. We'll have lots of things going on. So Joseph, lysogenic focused on replicating its viral genome, and lytic cycle killed the host cell. The main way that I see the difference is that the lysogenic cycle involves incorporation of the viral genes into the host cell chromosome, where it sort of can lurk there as what's called a prophage. And just to show you guys, I'm going to show you something that's really fun. So check this out. I have a song that is called I'm a Virus. And here it is right here. It's a really, really fun song. So I totally want to encourage you to listen to I'm a Virus. And also, if you go here, oops, I want you to notice that I have a whole tutorial about viruses. And there we are. And right here, mutation and viruses. And this will really teach you everything that you need to know about viruses for the AP exam. So that's pretty much it. And guys, yeah, so listen, you guys are awesome. This was a totally fun hour. Please, do your best, keep studying. And I'm going to see you here on Tuesday, 2 p.m. Tell your friends. This is Mr. W signing off. You guys are awesome. It's a tough situation that we got here, but we can do this. We can learn biology. I'm going to be right there behind you. If you want to give me some feedback about things I can improve, shoot me an email. You can do it from my website. There's that contact page. If you click over here where it says contact, you'll send me an email and I'll totally appreciate it. You guys are the best. So take it easy. Mr. W signing off. Stay safe. Practice social isolation. Be well. Bye.